Assalamu alaikum. Now I would like to talk about activation of helper T cell. This is the cell membrane of antigen presenting cell. This is MHC and this is antigen. So first signal is cell membrane of antigen presenting cell. In here there is MHC. It will process and present the antigen to T cell. This is T cell membrane. This is CD4. This is primer. This is present in primary signal or first signal, and the CD3 is also present. And this is T cell receptor. This is so it is first signal in T cell activation. In T cell, we know that there is helper T cell activation. There is cytotoxic. This is CD4 positive. That means helper T cell activation. In helper T cell activation, the primary thing that extracellular antigen, that means foreign protein, is phagocytes process as present on MHC class 2, which is expressed by this antigen presenting cell. And in here, in the other side, there is T cell membrane. In T cell membrane, there is a T cell receptor, and there is uh, additional CD4 positive, and there is additional CD4 cell, and there is additional CD4 cell, and there is additional signal is present in the name of CD3. So this is primary signal. Now this is first reaction. The second reaction, that from T cell there is CD28 and the B cell, and in the antigen presenting cell there is B7. So this is called co-stimulatory signal for activation of T cell, that B7 present on the antigen presenting cell, and CD28 from the T cell. Activate by combining these two signal, there is T cell activation, that means helper T cell activation. If there is and if instead of CD28, if there is presence of CTLA4, it means immunoregulation. If there is lack of B7, it is called energy, that means no immune response. If there is lack of CTLA4, if there is absent CTLA4, it is means no immune regulation. So it is first I have already discussed you about CD4 that positive, that means helper T cell activation. Now in helper T signal activation there is a signal 1, that means interaction between antigen and MHC complex, and second TCR and CD3, and the second signal is interaction between B7 on the antigen presenting cell and CD28 on the T cell, and I have already told you about. Now CD8 positive cytotoxic T cell activation. In cytotoxic T cell activation, there is perforin. This is T cell TCL, T cell receptor, cytotoxic T cell activation. There is the first two thing is same, that cell membrane of the antigen presenting cell process and present antigen to the T cell, and there is T cell with T cell receptor. And we know that when T cell, cytotoxic T cell gets activated, there is when there is activation of cytotoxic T cell, it will release some cytokines in the name of interleukin 4, 5, and 10. Interleukin 4, 5, and 10, and these cytokine will causes the release of this perforin and granzyme. Perforin will and the death will occur via two processes. By releasing of perforin and granzyme, perforin will create pores, and granzyme will enters into the cell and causes granzyme-induced cell death. And there is another process called fast ligand fast death receptor. Fast ligand present in the T cell, and fast ligand present on the T cell, and fast death receptor, that means CD95, present on the target cell. It will combines and activate apoptosis. This is second process, and this is how there is T cell activation. This is, I have already told you about the energy. Now activation of B cell. In B cell activation, the same thing happens. This is B cell on the B cell. In B cell, will process and present antigen with the help of MHC. With the help of MHC to the T cell. With the help of MHC, it will process and present antigen to the T cell. There is T cell and there is T cell receptor. In here, the secondary signal is CD40 present on the B cell, binds with CD40 ligand. CD40 binds with CD40 ligand, which CD40 ligand is present on the T cell, and there is B7 which is present on the B cell, and CD28 which is present on the T cell. By stimulation of by combination of these two signals, there is activation of B cell, and it will start releasing the cytokines. Cytokines in the name of interleukin 4, 5, which will mediate isotype switching, hypermaturation, and maturation of the plasma. Maturation of the plasma, and that is all about B and T cell activation. Inshallah, you will be benefited.